heart.
Hoje está presente em festividades culturais em todo o Brasil. E essa arte pode ser vista como uma prática de atividade física, trazendo melhorias no condicionamento e na coordenação motora.
Today, taiko is present in culture festivals throughout Brazil, and this art form is also regarded as a physical activity that enhances fitness and motor coordination.
O Abaredai é constituído por mais ou menos 15 integrantes, sendo todos voluntários, inclusive o nosso instrutor, Azumi Shiruji. E o estilo de percussão é diferenciado pelo fato dos tambores serem tocados juntamente com músicas acompanhadas por cantores.
Abaredai consists of approximately 15 members, all of whom are volunteers, including the instructor, Azuma Ishiruchi. The group's percussion style is unique, as the drums are played alongside songs performed by singers.
Tá, então eu vou apresentar nosso instrutor, Azumi Shiruji.
So he is the instructor, Azumi Shiruji.
Eu, Shirley Sato.
She is Shirley Sato.
Arlene Nakamura. Noriko Mizuta. Aí temos as cantoras.
And the singers.
Mihoko Maeda, Giovanna Yabe e Aline Kiminami. Espero que gostem.
Enjoy.
幸せなるなら二人してきっと前の時愛の垂れさず今母さんと話した女は夢そめ離れたから愛のそうさよなら言ったから寒い北の風は体壊さずしゃべった今思い出も写真一枚入ればいい
背中よでかかった赤いランドセル雨の中泣いていた学校ないよ夢だった20年ありがとう我が子長い旅疲れたら時々帰れ明日の朝まで飲んでるよ別れの朝だ起こしてくれよみんな思い出持ってゆけ写真一枚貼ればいい写真一枚貼ればいい
Hello, good morning. From the Japanese universities to come forward and take a photo, an official photo with the vice rector and local authorities. Please. favor. Paulo, por favor, também.
Só um pouquinho, peraí. Só um pouquinho, eu vou rearranjar os. Você vai.
Konnichiwa. Is that okay? Something I can't say. The other things I can say in English and Portuguese. I'm very glad to receive you. World. And we need to take care of these people that are hungry. So talking about animal and plant science and health, the genetic engineering from plants and animals' health, and the disease of plants and animals, we are going to have big explanations this day of today until we go to Londrina by the end of the day. So I thank you very much, and I hope you enjoy your stay here today. Thank you for being with us.
I also invite our provost, Dr. Mauro Cavalieri, to say some words, welcoming words.
Good morning to everyone. I would like to say that we were very welcome here in this rainy morning. I would like to thank your visit in the person of Professor Watanabe. And I would like to say that here at the State University of Maringá, we have two of the well-evaluated graduate program, PhD program in Brazil, in agricultural science. And we are very happy to receive you and to have the possibility of cooperation and collaboration in innovative research in this area. Thank you very much.
Professor Watanabe. Olá, pessoas. Welcome to our university.
Muito obrigado e muita companhia. Me agradeço por vir aqui em Maringá e outra vez muito obrigado à senhora de interesse e a consideração pela possibilidade de colaboração. Entonces tenemos unos pesquisadores aquí y quería introducir uno de ellos. Primero, profesora de cultura, por favor. Y segundo, señor Nagata de Japón. Y luego, doctor Donaka, Satoko Donaka. Y profesor Tamura de la Universidad de Kagawa. Señora de Barón Rivera. Aunque es de Filipina, él es mi estudiante, estudia para doctorado. Y luego señor Otsubo. Ingeniero, apóyanos respecto a la investigación. Señora Soya. Coordinadora, y cuando pregunta respecto a cómo envía estudiantes, ella encargo para coordinación. Y doctora de Asano. Y últimamente a doctor Kanamori. Doctor Kanamori trabajaba en Mondolina a largo plazo en el mejoramiento de soya. No olvido nada. Okay. Entonces, hoy día nosotros hablamos hablamos tanto, yo hablo español, pero no portugués, portuñol. Entonces, compartimos sus ideas de cómo mirar la posibilidad de área de investigación. Entonces, esta vez enfocamos área de agrícola y medio ambiente, pero luego, si ustedes tienen otra área de interés, podemos tal vez enviar a otros equipos a ver más posibilidades. Entonces, muy agradezco a tener ese tiempo y hablamos tanto hoy. Muchas gracias. Muy obrigado.
Thank you all. You can now have your seats. We can carry on our event.
Só pode vocês em pé. Só um pouquinho mais para cá. Obrigado.
Thank you. Carry on with our event. The audience will now split in two groups according to the topic of interest. In this room should remain those interested in functional foods. We're going to have 4 presentations related to this theme, coordinated by Professor Paulo Pinto. And those interested in animal and plant genetic engineering, inputs in agricultural economy, advanced diagnostic methods and techniques for animal and plant disease should move to the room at the end of the corridor, amphitheater number 1. You can follow our program in the booklet you have received. And last but not least, some housekeeping information. Sorry, we don't have Japanese tea, but we have Brazilian coffee at the rear of the room. We have water over there as well. Toilets are on the corridor on the left, on the right side, sorry. And well, I wish you a very good journey and nice work during the whole day. Okay, the Taiko group, please come forward to take a photo with the
Okay.
Só tá guardando o meu patamar, tá? Banheiro? É só para ver se não fica na frente do. Vai ser assim.
Não dá na frente, não dá na apresentação, tá perfeita na apresentação.
Não sei, vamos ver se fica. Fica melhor? Acho que podemos começar. So, good morning, everyone. Bom dia a todos. We are going to start our discussion this morning. I will just say a few words in Portuguese, okay? Then I will turn in English. Então, só para contextualizar, nós estamos realizando a missão entre o Japão e o governo do Paraná para possíveis colaborações. E nós estamos fazendo visitas em todas as instituições do Estado. No dia de hoje, nós estaremos aqui em Maringá e, explicando brevemente, essa missão ela foi dividida em alguns subtemas. Então, nós temos representantes tanto das universidades do Japão, quanto das instituições aqui do Paraná, que se alocaram em cada um desses subtópicos. Então, eu vou pedir brevemente, né, que eles se apresentem para que vocês conheçam quem são. Então, professor Jackson.
Português. Meu nome é Jackson. Eu trabalho na área de transmissões, do tema 1, que é seria
Professor Ricardo Ayubi.
Bom dia. Biotecnologia digital do Estado de São Paulo. Agora, é um prazer estar aqui com vocês, de Roberto e operações.
Professor Marcelo Canteri.
Bom dia, Marcelo Canteri, da Universidade Estadual de Londrina, já fui colega do Dauri, que está aqui na UEM, e do Ail, que está na UEPG, que trabalha com um aluno aqui, do grupo da professora Sheila, né? Professora Sheila, estamos na parte de automação agrícola, a professora Sheila mais na parte animal, mais na parte vegetal, e estamos à disposição para parcerias.
Professor Selwyn.
Bom dia a todos. Sou de Sábado, sou em patologia, aumenta-se o diagnóstico de doenças de animais. Não qualquer doença de animais.
So we just did a brief presentation from the researchers of Paraná State. So we are going to start our talks. We are going to have 4 talks this morning. I'm going to be the first. I will present briefly what we are doing here at Maringá State University. And then we will have other 3 presentations from professors from here. Professor Ricardo Vasconcelos, Professor Silas, and Professor. And then at the final, we open to discussion. Okay? So I'm going to start talking about precision livestock farming and how we can use it for some tools for both to study animal physiology and then to apply on animal welfare. So here at State University of Maringá, I coordinate a research group, and I have some of my students here. So please, I have graduation and undergraduate students, basically from animal science course. And our group studies innovation and sustainability in animal biometeorology. And we are, for 4 years, studying this here at UEM. So I bring here to scan if you want to see our Instagram. And I will present some points about our researches. So the main problem we have to face nowadays is about climate change and the rise of the temperature of the planet Earth. So we have some modelings that already describe we have the temperature more than 1.6 degrees higher than what was expected in the last years. So we have to bring this concern together with increase of solar radiation, mainly in tropical areas like Brazil. Also, when we see the scenario from Brazil about climate change, we have also some concerns because we can expect until 210 the rise or the increase of temperature in all Brazilian biomes. And we have some concerns about salinization, mainly on Amazon, and the increase of rain in some other places, like the south of Brazil, like we are. And this also, we saw, I don't know if you guys in Japan could see, but last year we had a flood, a terrible flood in the state of Rio Grande do Sul. And also, we have the increase of fires, the increase of drought in some areas, and this could limitate the Brazilian agriculture potential. So we have to consider this also for animal or livestock production. But Brazil is one of the main producers. We have, I tried to update these numbers, but we have more than 250 million cattle. We produce almost 34 billion liters of milk per year, and we have a population of broiler chicken around 1 billion animals. So just in Cascavel, that we were yesterday, they have more than 21 million broiler chickens. So it's the city with the most number of these animals. But for Brazil, maintain this position, we have to consider the impact of climate on these animals. Basically because we hear these animals in free-range areas. So they are exposed to mainly high solar radiation. So in our group, the main question we do is how can we continue to be one of the world's largest producers of animal protein. So we try to develop our researches using physiology studies linked to animal behavior. We try to do an evaluation of the meteorological conditions these animals are exposed, also to evaluate the condition of animal welfare, and then to study the thermal equilibrium, how these animals respond to this climate. And all of this, we use the precision livestock farming tools to this monitoring. So basically, the main research lines we do is about image diagnostics using thermographic cameras. We also use artificial intelligence and the Internet of Things. So basically to monitor climate, physiology of these animals, and then to apply on animal behavior and health. Also, all this data collected, we generate big data. So we have also to work with all these informations, basically trying to identify some patterns and also to develop some models from this. And the other is about the use of computer vision. So we try to estimate, like for example, identification of animals, to study some behavior of them, and to identify pain and stress of livestock animals. So I try to bring the main lines we are doing here. You saw at the presentation, we have an experimental farm here, but it's along 15 kilometers far from this campus. So we developed our experiments, some of them there, and some of the experiments in other places, like producers' farms. So this was an experiment we did with dairy cattle about the evaluation of some shading structures. So this is a material used for greenhouse, and this is from Japan. And we tested this net for shading resources for dairy cattle. And we also always try to bring some answers, mainly for small producers. So for those who don't have so much money to invest on their properties. So the use of nets is a cheap way to protect animals. And we evaluated this net from Japan and a combination of a black and a reflective material. So both of them was okay for the animals, and we find mainly studying with the livestock precision tools, we found a decrease of body temperature of these cows and also the respiratory rate of them. Also, we found lower mean gradient temperature and the soil temperature. So the animals use to lay down in the soil. So it is a reliable indicator that the comfort is okay for them. Also, we do an evaluation of some alternative covering materials. Also think mainly in small producers. So we use milk packaging. So we collect this material and transform in the use for cover of the installments. And we are doing some experiments in minor scale prototypes. So we are trying fiber cement tiles along with the use of milk packaging to reduce the temperature inside the installments. Other work we do is about the study of some mobile devices for the monitoring of animal welfare. This also connecting some Wi-Fi sensors to monitor in real time the condition inside the installment. And we did a first trial to connect the fans also to these sensors. So trying to do some automation of the installments. We also do here using infrared camera. So we do, or we did an evaluation of the thermal comfort zone for broiler chickens, also studying some behavior and the thermal equilibrium of these animals. So trying to find what is the correct thermal neutral zone for these animals in all the states, in all the hearing states of them. Also, we use infrared thermography for the evaluation of feedlot systems. So in Brazil, we have big feedlot systems with a lot of animals and little resource for them, mainly to avoid the high solar radiation. So we use it to evaluate the comfort and the condition, the body temperature of these animals under the systems. So you can see here, right? Very high temperatures, the animals with almost 53 degrees Celsius. Also, we apply for pasture systems. So we use the same tool to evaluate dairy cattle and to estimate body temperature and some behavior answers of these animals as well. And we try to apply in some image analysis. So we try to find some patterns and to correlate with stress and comfort conditions of them, and also link it to the color. So if the cows have most of body surface black or white, and we correlate the search of these animals for shading resources and try to evaluate using image analysis like this. Also, it's not the main purpose here, but we also do for some conservation areas. So we have a study in Ingapar, which is a conservation area here in Maringá. And we are doing also a study to evaluate the welfare of turtles who live there in the river we have inside the pond. So also being a non-invasive tool, and we can evaluate the condition of these animals in the systems. And for last is the trial we are starting now. in a producer farm, in a compost barn system in the state of São Paulo, but it's here near Paraná. And we are going to evaluate 200 dairy cows, mostly dairy cows, and we are going to use collars to monitor 24 hours the behavior of these animals. And we are trying to connect some methodology to measure the body temperature of them. And also, on this project, we are going to correlate the incidence of some heat waves on reproductive answer of these animals, productions, and also behavior analysis. So we are going to use another sensors like this to measure the vaginal temperature of them in real time. We use some sensors or equipment to monitor the microclimate condition inside the barn of the cows. Here are the collars, which we are already using and monitoring. And here is just an example. We can follow up all the time the animal spent ruminating, the animal spent on rest, if the animals had some painting, and the time the animals, for example, was walking in the barn. So this is an example of a project we will have a lot of data, and after then we have to analyze and predict some models from that. So it's our project which we are starting right now. Also, we do some observations in feedlot systems using drones to monitor animals. So here is just an example of some photos we did, but we use the tool for counting the animals. Some tool to evaluate the shade we have for them, and we use the drone to evaluate the structure of the place the animals are, like if there is some problem in the water trough, we can see rapidly and have a decision making, and also to monitor the feed of the animals. So it's a very important work as well, because here in Brazil we have feedlots that have more than 50,000 animals. So only a person doing this monitoring is really difficult. So the drones is validated for this, and also coupled to monitor some behavior and physiological answers of these animals. So I put here what we can do from this, some ideas I had when I was doing the presentation, just for our discussion then. But we can apply other sensors to be more valuable, the information we collect. We can also develop other sensors and tools for the monitoring of the animals. We can, and we have to do, is how to analyze this big data collected. Also, the development of some prediction models. So we have a lot of data from all these researchers, and we have to do the modeling of them. And also to apply to other animals, because we work with all the livestock species. So we can apply to swine, sheep, fish, and etc. And I put the etc. there because we can discuss other ideas and how we can follow up what is here. So my presentation was that. Thank you very much. So now we are going to have the presentation from Professor Dauri.
Good morning, everybody. Thank you very much for your visit to our university. Thank you very much for the organizer for allowing my presentation. In the last years, maybe 15 years, I have done this research working with fungal plant pathogens associated with mycotoxic problems. So the species of Fusarium and their associated mycotoxins affecting production of wheat, barley, and maize in Paraná. Our major goal is pursuing quality in the products. So just for organize our location, we are right here at the moment under the Tropic of Capricorn. It's a line that divides the Brazilian agriculture in more subtropical to tropical. This is the red soil of this region. This is it's the Paraná state. I could put a line here. Here is cool, here is hot, and this red soil. And down in the south, the soil is more acidic, and the weather is cool. So the disease I have worked is in wheat and barley. Fusarium head blight is a worldwide problem. Because this disease has some mycotoxin associated, deoxynivalenol, nivalenol, and zearalenol. In Brazilian regulation, we have standard maximum limits for deoxynivalenol and zearalenol. We don't have limits for nivalenol, but some industries here in Brazil, they have some industries here in Brazil, they require more quality even than the regulation. And in maize, the disease are this stalk and ear root caused by Fusarium. And the mycotoxins, we have one more, fumonisins, is also is regulated. In wheat, it's about the grain, it's 3,000 ppb. And in maize, fumonisine is 5,000 ppb. The flour is 750, 100 ppb. So our regulation on mycotoxins is more close than the European regulation. The American regulation is quite different, but we approach the European system. We have a very, I would say, very modern and effective regulation on food quality regarding to mycotoxins. So all these mycotoxins, my approach has a pre-harvest mycotoxins, the mycotoxins that contaminate grains in the field. What we can do for reducing this problem? Okay, the questions we have pursued is which Fusarium species are responsible for this disease, since Fusarium have different mycotoxin profile. Which mycotoxins are potentially associated with this species, and which are the impact in quality, and how we can impact mycotoxin, minimize. So where are the wheat production in Brazil? It's mostly in the south, and we are moving to the center here. The last frontier of Brazilian agriculture is the savanna area. Here is the Amazon region. It is the savanna area. In the last 30 years, a lot of people moved from south to savanna area for planting soybean. Even Japan started a project in the 80s, very important for Brazil start colonization. Paulo Centro was the name. And most of the wheat produced in Paraná and Rio Grande do Sul state. Another thing, okay, we plant wheat in the spring. Spring is for in southern hemisphere. Is from spring to is from autumn, winter to spring. So we have a double crop system, winter and summer. Wheat is a winter crop. Soybean is mostly our major summer crop. And our system is, I would say, the majority is a no such system. And so this is a typical in the southern Paraná, it's in the northern part. And barley. Okay, let me stop. Brazil produces about 40% of our needs in wheat. But Brazil is a considerable exporter of wheat-based products like cookies. So we import wheat, but we export wheat products. But in overall, we have about 40% of our needs. We import wheat from Argentina, Canada, and other places. About barley, mostly our barley is basically for malt, for brewing industry. Our production is not so much. We are big importers of malt. The area is increasing. The malt industry in Brazil is increasing. And in the malt industry, the part of it is used also for animal feed. Okay, and maize. Maize is cultivated in all entire Brazil, basically. Paraná is a big maize producer, and maize is produced in Brazil in 2 seasons. We have a summer season and an autumn-winter season. Like here in Paraná, here in this region, now we have a lot of maize in the field. Maize, and you have a little bit of wheat. And in southern Paraná state, maize is a summer crop. Okay. And about production, Brazil is an exporter of maize. We are the 3rd major maize producer in the world after China, after the United States and China. Okay. Our mycotoxin targets have been the oxynivalenol, which is regulated. We are keeping eyes in the acetylated forms, 15-adone, 3-adone, and also nivalenol. And so these toxins are produced mainly by the Fusarium graminearum species complex. So far, we didn't have Fusarium humorum. And we are keeping our eyes in the type A trichotoxin, 90%, is T2H2, and the PAS, in these novel toxins, and also zearalenone. So, just for refresh, what we call it Fusarium graminearum some years ago, now is considered a complex species. It has about 15 different species with different mycotoxin profiles. So in Brazil, we have Fusarium graminearum sensu stricto, Austro-Americano, Cortaderia, and Meridionale. This, there is one Brazilian, this is from wood, it's not from tree. And this complex species can produce different mycotoxins, like we call 15-adone population, when it produces a lot of DON and a little bit of 15, the acetylated form of 15-adone. So far, the regulation still doesn't count 15-adone amount. We call the population 3-adone when it produces DON, and 3-adone also is not counted. Then we call nivalenol population, that Fusarium species that produce only nivalenol. And now we have this toxins in I guess some parts in Europe. Okay, so what we have in the last years is collecting isolates. First question is understand which fusarium species we have. We need to know for predict the mycotoxin problem, and also for composing the inoculum when we do inoculation for genetic resistance, fungicide testing. We need to know which fusarium we have, okay? And we have done this using multiplex PCR and this PCR Luminex approach. Right now, I'm no longer working with Luminex. I have sent it to the USDA in Peoria, Illinois, for this. The advantage of PCR and Luminex is it's fast. You can do 100 questions at the same time. So then we can separate the populations. We have some worries. First, is like 10 years ago, we had some questions in our mind. Which would be the relevance of vivalenol in our production system? Which would be the relevance of 3A-DON population in our production system? Because the 3A-DON population is supposed to be more aggressive and more stronger DON producer. So we did this study. We published in plant pathology. This Carolina Pereira was my PhD student, and we assessed 754 isolates from South Paraná, and to have a picture of what fusarium we have. And the majority is the Fusarium graminearum species complex, and inside this Fusarium graminearum species complex, the predominance is Fusarium graminearum 15A-DON. But we have also the second place is Fusarium meridionale. This is a potentially vivalenol producers. So 10 years ago, I was thinking, hey, how big is our vivalenol problem? I know that in Japan would work vivalenol. But the Americans, they don't have this problem in the wheat production system. They have a little bit in the south. In Europe, they have vivalenol. So with this, that helps us to define the inoculum, because we have a partnership in southern Paraná with the Cooperativa Agrária. They do screening for genetic resistance, and we needed to define which inoculum we use for inoculation. So, another, we describe a new species of Fusarium, but it's a secondary. There is no relevance. We published this in mycology, also this PhD student, with the people from Peoria, Illinois, it's the big guy of Fusarium world. But it's a secondary species. It's not so much aggressive and do not produce relevant mycotoxins. Some years ago, we assessed the Fusarium graminearum species in maize from south Brazil. We found that in maize, Fusarium meridionale is more relevant than graminearum. So it's an interesting finding. And this is another PhD thesis out of Texas from the Cooperativa Agrária at the time. We analyzed sites so far are part of our management. Our weather is very humid for wheat. So we needed to consider that we are producing wheat in a region with sometimes 2,000 millimeters per year. And we are producing wheat in a very wet, under very wet conditions. So fungicides are part of our strategy. With this work, it's 4 years work, we published in crop protection. Based on this work, we decided no longer use distributors after flowering. only 3 years old. In a fungicide approach for controlling weed disease, we have a problem because the big driver of the industry in Brazil is soybean. They start to think only about soybean. We don't have the same formulations like the Americans have or the Europeans have. Because here, the big companies, they try to push their formulation from soybean to wheat. So that we have some problems with this. This is another work some years ago with my colleague Julian Almeida. We did a screening of different wheat cultivars for resistance to DON accumulation. So far, our approach is to combine the best resistant varieties available with the best timing of fungicide spray. So far, this is what we have done. This is another work we published more recently. When we test, just for having an idea, the different timing for inoculation and fungicide response. So under our conditions, we needed to work with multiple sprays. And that we needed to optimize this issue. We have it's not an updated assessment, but some years ago, we evaluated about 700 isolates from Rio Grande do Sul to the Savannah area. And we reached the conclusion that our major species is Fusarium verticillioides. We have a very few subglutinans, Epiceti, and proliferatum, like Argentina, they have proliferatum. The verticillioides and proliferatum are fumonism producers. So what conclusion we reach that overall in Brazil, okay, we have Fusarium verticillioides, which is potentially fumonism producer. And in the south of Brazil, we may have the Fusarium graminearum species complexes, because here we have more wind. It's cooler than the northern part. This assessment needs to be updated. Okay, so I would like to highlight my research partner is this Agraria Cooperativa in South Paraná. Is the biggest malt producer in Brazil and a strong wheat producer. They have clients like Nestlé, this big company with high quality standard. They have ISO 22000 and so and so. And my partner is there is Dr. Julian Almeida and Alfred Stutzer and Miriam Tuzupiazzi, that is the barley. They have a strong central lab with this UPLC equipment, and they conduct a lot of field work. So the next step, what do we need to do? We needed to continue to proceed our monitoring of Fusarium species and trichothecene genotypes, Titinagon, Triodon, and Nivalenol. Understand if there is any fluctuations. One big question is the 3A DON population could increase in time and predominate. The Nivalenol population could increase and dominate. We needed to answer this question. Another issue is a new toxin that the Americans describe. So far, we don't have any evidence that we have here. We needed to monitor the type A trichothecene. So far, we presented a paper in the last Fusarium Head Light meeting in Austin, Texas, in December. We have a 5 years assessment. We didn't find so far this type A mycotoxins, but we need to pay attention to this. We need the surveillance on Nivalenol relevance in crops. In this sense, Japan has experience. I have cited that papers, very nice papers, important papers in this issue. We needed to update our diagnostic and identification tools. We need to enhance, improve mycotoxin management practice through genetic resistance. We foresee that the genetic resistance has an important role in this management of the mycotoxin. Fungicide formulations is always important tool, and we needed to check the quality and so. And more recently, we have thinking more on developing strategy for managing fumonisin contamination in corn. So this is my presentation. Yeah, what I have done in this slide. Thank you.
Thank you, Professor Dauri. I will now ask Professor Ricardo Vasconcelos.
Thank you, Sheila, for the presentation. It's nice to be here with you, and it's a pleasure. And I work in a different area because I'm not working in animal production, but with dogs and cats. And my expertise area is in dog and cat nutrition. And I will present to you here some aspects before related to our graduate program here, because I'm assistant coordinator today, and I will present some aspects related to our structure. I will present our research lines, mainly the main lines related to the dog and cat nutrition that I'm working here with my students, and also about our laboratory structure that can be interesting to. Okay. So. So I'm a veterinarian, graduated at Federal University of Viçosa, and I'm a specialist in dog and cat nutrition. My Master of Science and PhD was performed in São Paulo State at State University of São Paulo, and today I'm working here in the Department of Animal Science since 2012 as professor in dog and cat nutrition and animal physiology also. So our animal science graduate program is a program that started the activity since 1993, and we graduated more than 900 Master of Scientists and PhD that are important for our region in Brazil, and also many professionals are working abroad and also in other states in Brazil. And we are today in 18 professors, and I am today, my position here is as assistant coordinator of our program. And as I told you, the main focus of our program is in animal production. But we have some different areas here because some students need to want to become professionals in different areas like pet food production that I'm working right now. Brazil is the third biggest pet food producer worldwide. And the pet food production is important because we use many residues from the human food production. And the pet food has an important position in this aspect. And my research area is focused on the circular economy of ingredients and other nutritional aspects. So about our structure, we have some facilities for in vivo studies here. I don't know if they know, but Did you list it? Okay. After here, you will visit our structure of laboratory, but we have 2 farms, and one of them is placed in Iguatemi, 15 kilometers far from here, and other place called Codapar that is placed 20 kilometers far from here. That is specific for aquaculture production, especially in tilapia production, that Brazil is an important producer in this area. And in Iguatemi farm, we have some facilities for cattle, beef cattle and dairy cattle production, swine, rabbit, poultry, chicken, goat, sheep, whale, fish, and other species like this fish that I'm working that is catching. Okay. And about our lab, we started the activities in 2014, and it was possible because we have some partners from the suppliers of additives and ingredients and pet food companies. And it was important to get funds to start our activities in the research. And today we have a small facility for cat nutritional studies that we can study some support the pet food industry with some tests like digestibility, palatability tests, and urinary pH that is very important for cats to avoid the blue lights. And also we study new ingredients and other aspects related to the cat nutrition and health. Okay, and we have about 30 cats there. And this year we are studying to remodel this facility to improve the quality of the structure for the students and also to support the pet food industry in Brazil. And the main focus there will be to develop in vitro methods because replace the use of animals is important in our area and other animal areas. And we are developing some methods to study digestibility and fermentation by in vitro approach. And we are studying also some aspects related to developing new ingredients from residues from the industry in the Brazilian scenario. And also we are developing some studies at home with the owners. And this is important, especially for the pet food companies, because some tests like digestibility and palatability, we can provide this test in almond animals in real conditions, and we are validating this method by the results that we have in our lab together with the owners. So we have other structure here related only for the study of quality of poultry byproduct meal and fish meal. About 40 to 50% of the animal after slaughter is considered a residue for human consumption. And so pet food sector is very important in this aspect because poultry byproduct meal, fish meal, meat and bone meal, and other animal meals are used for the production of pet food, like as protein sources. And we have many problems with quality of these ingredients. Instead, they are very nutritional important for cats and dogs and other animal species. And so we have a small structure here that we can produce small batches of these ingredients. And we are focused on, especially in quality of poultry byproduct meal, fish meal, shelf life of these products, because we need to stabilize them for, especially for oxidative degradation and also microbial growth in these foods. And so we are testing some additives for preservation of these ingredients. And also, as these ingredients are produced from fresh meat or fresh residues from the food human production, if we wait many time to process these ingredients, we can have some problems related to the contamination of biogenic amines, for example. And we are studying some, and Brazil is a tropical country with very high temperatures, and the degradation of this material is very important. And we need to study methods to process these ingredients as fast as we can, of course, but by preserving these ingredients with different additives. And today, customer and pet food companies are searching for natural additives, and we are studying some different natural additives produced in Brazil and other commercial additives produced abroad. About our structure of laboratories, we have the main laboratories that we use to support the sample collection at farm, at the cattery, and also in our pilot plant. We use it. We usually analyze these samples here at the State University of Maringá, here where we are right now. We have 4 main laboratories that we use, that are laboratory of animal nutrition here in the block, in the neighborhood here. The laboratory of for in vitro studies, that is near here also. The multi-user central laboratory that you are going there to visit it, and the laboratory of chemical analysis and water and foods that we use this lab a lot for chromatography analysis, especially. Okay. The laboratory of animal nutrition is working since 1980, because the animal science department started the activities before 7th year, and we have this lab from that time to now, and this is the main laboratory that our students is working today, and we perform there more than 5,000 attacks per month, especially related to the thesis and dissertations from students and undergrad students that use to learn methods and others. And we have there 3 laboratory technicians, because they need to give support for students in the learning process. The multi-user central laboratory is the most important complex in our university, and there we have a lot of equipment that you can see after our meeting here. And the Department of Animal Science, Agronomy, and Veterinary are responsible for the CAA that has approximately 30 equipments, but the equipment aren't there. They are distributed for the different laboratories and farms, because our experiment, we need to collect samples in practical conditions and analyze immediately after. Okay, and we have there 9 central labs that you can see there in your presentation. About the laboratory of chemical analysis of water and foods. This is an important laboratory that we use in our research, especially because chromatography analyses are very important, related to the contaminants, additives, and nutritional aspects related to pet food production. And in this lab, we have this list of the main analyses that we perform there, that are biogenic amines, synthetic antioxidant residues, minor reaction products, volatile compounds related to the smell, flavor, and also oxidation in pet food products and ingredients for animals. Short-chain fatty acids, long-chain fatty acids, and other analyses, especially related to the natural antioxidants, essential oils that we are working also in this area. Okay. And in this laboratory, we have these equipments, ultra-high efficient liquid chromatography, high efficient liquid chromatography, gas chromatography, coated to mass spectrometers, and others. And we have there a technician that is responsible to support us to develop new methods and also to attend the industries and other users to develop new methods and perform the analysis. Okay. And about our laboratory specifically for in vitro digestibility and fermentation is placed here in a block here near here. And we are developing some methods to replace the use of animals, especially for digestibility studies, and to evaluate the effect of some functional foods in the fermentation microbiome of animals in a faster way than the use of animals for experiment. That is very expensive, that sometimes we compromise some aspects related to the welfare. And so the development of in vitro methods are very important, but it's a challenge, of course. We have also a structure to extend the shelf life of pet food and ingredients. All of the ingredients that we produce here for research, we have some structure to evaluate the stability, oxidative stability of this food and microbiome growth during the shelf life. And also we support some pet food companies in Brazil that send to us the samples that they produce to analyze the shelf life for exportation and to sell these products in our country. About the in-home methods, this is a new subject in our area, and we have some examples in Europe, for example, like Pet Panel, that is a company specialist to support the pet food industry in the in-home panels of animals. And this is important because by developing methods to analyze, to evaluate the pet food products developed by the pet food industry, is important to replace the use of animals in laboratory conditions and use these animals in outdoor conditions. But of course, it's a challenge because we have many aspects that can influence the results of these methods. So we use our structure to analyze the method in the laboratory, and after we compare with the in-home methods to validate these methods for palatability and digestibility especially. Here we have a partner from the pet food company, and today we have this signature of this software available for use. Simapro is a software developed by in Europe, and that is very important because we have an extensive data bank about the life cycle analysis or environmental impacts of different products from different sectors, and we can use this software today to develop to calculate the environmental impacts of different ingredients and products developed developed for pet food sector. But of course, we have many data that related to the soybean production, maize production in Brazil and other countries that are very important to analyze. Especially today, we are worried about the carbon footprint, but of course, this software is very important to determine the environmental impacts of the production of different ingredients that companies today are looking for, developing new products related to the less environmental impacts. And this is the way that we are studying today. And of course, we are studying new ingredients and calculating the carbon footprint of these new ingredients, like derivatives from algae, fungus, and other different ingredients for pet food production. And here we are, it's me and my students, and we are about of Science and then PhD, and me as advisor here in our department, and we work with some colleagues from other departments and other areas because it's very important to develop our sector in pet food production. Thank you.
Thank you, Ricardo. Now I will invite Professor Silas for his presentation.
Hello, everyone. Thank you for being here today. I'm a Dr. Silas Marcelo de Oliveira. I'm crop scientist here in Maringá. I've been working with soybean, corn, and sorghum for the last 10 years. And today I select one of your recent works. Okay, it's not published yet, but we talk about a little about the soybean and nitrogen fixation. I present a little schedule for the next 10 minutes. We talk about how important soybean in Brazil, briefly, very short, why we use pesticide with seeds, result of our students and further students we have intended to realize. Okay, how important is soybean in Brazil? For 4 years ago, Brazil became the most important soybean producers in the world. Soybean is one of the most important brand and source of protein in oil to the world. And Brazil is the first one and represent about 35% of soybean in the world. Okay, so the most important producers. Okay. Another fact, and few people outside of Brazil know, Dr. Dauri talked about a little about this. So we have a very different scenarios for agriculture in Brazil. We have a tropical scenarios and have a subtropical scenarios. Maringá, the city where we have today, is a merged area. Have a transition because we have some characteristics from tropical, we have some characteristics from subtropical. But what's the big difference between tropical and subtropical for soybean, for corn, for cotton, and other crops? You have two big differences: temperature and precipitation across the year. When you analyze the tropical conditions, you have from April to August, you have a little rain. Some places you have no rain. Okay, when we come for the north of Brazil. When you go to the subtropical areas, you have we can say more distributed rain across the year. You have during the autumn and the winter, you have low temperatures, and these are we have to have subtropical climate. For example, we have no conditions for corn and the autumn in the winter because it's too cold for corn. In the other sides, you have, when we talk about tropical agriculture during autumn and winter, most of the times you have corn, like as second crop after soybeans, okay? Of course, this affects how crop, which crop we can talk about. For example, when we can about tropical, most of the time we have soybean or cotton, a small part of cotton, followed for corn, cotton, or grain sorghum. When we can the emergent area where I have today here has soybean and corn and grain sorghum. When talking about subtropical, most of the time it's have during the summer corn or soybean. Okay, it's a very different scenario. Followed for wheat or cover crops or other crops which can be cultivated during the winter. So what's our scenarios to talk about specific about the importance of nitrogen fixation for soybean? Most of our soils is lichen soils. Our soils fertility score have low tax of soil organic matter. Okay, we have exceptions for Guarapuava, for example. Okay, it's a very different scenario for Brazil, for most of the parts of Brazil. But our soils is a big challenge for agriculture in Brazil. If you come back 40 years ago, soil fertility was a big challenge for Brazilian farmers. Okay. Second important question: we have no credits for nitrogen. Most of the time, soybean is perceived that it's fallen corn, it's fallen wheat, and have received with highest carbon and nitrogen relationship. Okay, it's a very important scenario. So we have no low soil organic matters, we have no nitrogen credits, and most of the times when soybean is fallen for corn, for example, this is a scenario. We have no residues here. It's very difficult for soybean. It's a very challenging scenario. Okay, and with this we have a high nitrogen demand because soybean. It's so important for the world, it's so important for Brazil because one of the most important characteristics of soybean is the amount of nitrogen we have in the plants. We have it's a medium about this the soybean producers, but each tonelade of soybeans demand around 18 nitrogen kilos. Okay, if you produce in Brazil, the average of producer here is about around 4 tons. Okay, something like this. If you consider 18 kilos per ton, we have almost sometimes half ton of nitrogen for hectare. It's a lot of nitrogen, and we need to consider it. You have lichen soils. You don't have nitrogen credits from previous crop. Okay, and you have a huge nitrogen demand. So for us in Brazil, in crop science, it's a big challenge. We remain soybean, remain nitrogen free soybean. We provide this nitrogen. Most important source of nitrogen for soybean is not the soil in Brazil, it's the nitrogen fixation. So it's a very important topic for us because when we talk about how important it is in Brazil, my question is connected with this is how important is biological nitrogen fixation in Brazil. BNR is very important for providing soybeans in our scenario. Okay, so why use pesticides in Brazil? Why use pesticides in seed? Use of pesticides, it's a very polemic, it's very, how can we say, it's a complicated, okay? But why Brazilian farms use pesticides? Okay, you have a tropical and subtropical conditions. In this condition, it's very, very, very good for soybean, but it's very, very good for designs, for insects, okay? So we have a lot of problems about these designs, a lot of problems about insects. So to protect our plants in this phase, okay, we need most of the times use seed treatment with chemicals, okay? We don't have official numbers, government numbers. But when we talk about the farmers in Paraná states, in Mato Grosso states, in Rio Grande do Sul states, in different states on where soybean is very important, I am sure more than 95% of farmers use seed trains. It's very common in Brazil because we need this most of the times. Okay, so how much does the pest and the disease control for soybean cost? How much cost it is? If we take a price from Paraná states, it costs around $120 for hectares. Brazil in last year produced 45 million of hectares for soybean. If you take this picture and have a multiply colors here, you take around $5 billion for control of disease pieces in Brazil. It's very important amount for trade here. So we talk about two important things about crop production soybeans, okay? And we need nitrogen fixation and we need most of the time seed treatment for save our plants in the starting of the growing season, okay? So we have big challenge here. The study we're talking today with us is a study, most of the times, the Brazilian farmers inoculated radiolizobia, it's most important microorganism for providing nitrogen fixation. Most of the times, we inoculated seeds, soybean seeds, in the moment of the planting. But we have different scenarios across Brazil. Sorry. Most of the times, we have planters. Imagine a planter here, okay? Most of the times, we have planters with around 15 rows, 17 rows. It's a large planter, okay, machine. Most of the times in these farms, we have no conditions for inoculated soybean seeds in the moment of the plant. So how research works to help our farmers? One alternative strategy is we inoculate soybean seeds before planting. Sometimes industry have been working to inoculate soybean seeds 16 days before, 20 days before, 100 days before, together with seed treatments. But we know, and it's proved, that when you mix radiolizobia and chemical products, you have sometimes a penalize. Radiolizobia is it's life, literally, okay? So what's that challenge here? For big farms especially, inoculated before the planting is very good because the logistics, okay? And have some farms in Brazil have 50,000 hectares, okay? It's a big challenge for planting soybean, corn, cotton inside these farms. So this technology is not used for everyone, but for a very important percent of the farmers in Brazil. So radiolizobia and pesticides remain together sometimes during 20, 60, and 100 days before the planting. And we investigate the data I will share today with you for investigate how this affects soybean, how this affects the inoculation and the nitrogen fixation. Okay. To provide this data with best practice, we performed in the last years 6 different sites with different conditions and soil very, very different to know how this affects because of soil organic matter, because the date planting matter, a lot of things matter of this. So we have basically we have some controls that include nitrogen. Okay, some controls have branch dissolving, some controls don't have branch dissolving. And have a lot of data. I don't talk about each site specific, but we have an overall picture. Okay, when talk about the overall picture, what first compare I will share here is when we compare the traditional inoculation, the day of the plant, and the inoculation 16 days before the planting. What's was the first thing about yield. 4 of 6 sites we investigate have sustained or greater yield compared with traditional inoculation. It's very good, okay? But 2 sites will have a great yield when the inoculation gradually resolved in the day of the plant, okay? What's the point here? In some scenarios, have a huge difference between previous inoculation and inoculation the day of the plant. Okay, one of our challenge now is it's cover why. What's the difference between these results in green, these results in red? Okay, so when we think about the nitrogen fixation, one of our evaluations is counting the number of the nodules in the weight of the nodules. Some sites have difference, have a best performance for inoculation the day of the plant. In the other sites, have similar performance between treatments with different chemicals, okay, and control when you inoculate in the day of the plant. It's very clear for us that rain affect performance of this. Temperature affect performance of this. Type of the soil affect the performance of this. Okay, because when we work with brandisóbio, before beside the interaction and the chemicals and the brandisóbio, these conditions is very important. Okay, we don't have how answers here. This is a beginning stage to we know about this. Okay, and some highlights I would share here today. Pesticide combination affect yield inoculation. We know some chemicals that have been sold in Brazil have more impact, and the other chemicals have less impact in soybean and brandisóbio and consequently nitrogen fixation. Soybean seeds inoculate 76 days before planting, increase yield from 28 to 192% relative control, with no nitrogen and no brandisóbio. Okay, consider the best combination of the pesticides to brandisóbio. Soybean seeds inoculate 60 days before planting reaches similar or great performance in 4 of the 6 sites. This for us is very important because it's very common when talk about the farmers. The question arising for the farmers is this work? Is it this work? I can inoculate soybean with brandisóbio previous 20 days before, 30 days before, 100 days before. Okay. Soybean seeds inoculated 6 days before has a great potential for the farmers, but where we need to pay attention, we need to be more patient because we know have different things happen here. We don't have how answers were explained it. Okay, in Brazil, in last 5 years, have a huge demand for the other microorganisms. Okay, different microorganisms. One most after brandisóbio, I think the most used microorganism in Brazil is Azospirillum, including soybeans. We have a lot of studies, but how we have, for example, leaching soils with low, very low depths of availability of phosphorus. We have different microorganisms for works about phosphorus, potassium, and the other nutrients. So today we have a demand for knowledge about different microorganisms in different aspects in how these affect DNF, how affect soybean. This topic is very important for us because in the last years, I don't read it here today, this paper, but we published a paper 2 years ago, and we can prove across the years, soybean nitrogen concentration into soybean grains has been low. We lost nitrogen into soybean grains. It's a fact. It's happening in Brazil, it's happening in Paraná States, it's happening in the United States, it's happening in Argentina. Okay? So it's a challenge for us remain nitrogen concentration into the ground. For the guys from the animal science, it's very important because when they formulate the food for the animals, we have a very key difference between soybean, how can I say, loss. Okay, soybean from Paraná have X% of protein. Soybean from Mato Grosso have a different percentage of nitrogen and protein. It's very complicated for the industry. So we need to work to increase yield, but to keep soybean quality and keep soybean grain quality. Okay, 10 minutes, I think, probably, yes, it's okay. It's very short message. We are glad to receive everyone here today in our viewers, in our home, okay? We hope you enjoy Maringá City and enjoy University State of Maringá. Okay, thank you.
Yes, we are open for discussion now.
Let one respond.
Okay. The first one is there are results mixing value resolved with azospirilo. You have something about that?
Yeah, I have previous studies between azospirilo and resolved, not this one, okay? But what's the results of think about yields? The yield is increasing, but the tax is very low. We talk about between 2 and 5%. Recently, guys from Embrapa, from Londrina, published meta-analysis, and the results is very similar between these. 2 and 5% more yields when you use azospirilo. Okay, but what that concern us? When I share with you 3-4 different kinds of microorganisms, and when they put this together, that's dissolved. Put this together azospirilo, you really don't know what's happening. Think about yield, think about nitrogen fixation. So sometimes I have studies from greenhouse, okay, glass house, but it's very different when we can produce field. And for us, it's a challenge because the guys of the fazenda, the FEI, here. But for example, our experimental station in Guatemi had a kind of soil very different from the other places in Maringá. Okay, so when I have a result there, it's complicated. You can, I have these results, so now I can apply this for everyone. So most of the time I need 4 sites, 3 sites, 6 sites, and these results a lot of work. I have 2 undergraduate students in here, okay, Luisa and Gianluca, and they starting now, but if you talk with the graduate students, you can see have a lot of work to have these days.
We have good results when you doing a spray application of only azospirilo. Yeah. And you have better results in.
No, when some guys now have been working with brachyzobium spray and field spray. So what you have results start. One challenge for us is most of the time we have no irrigated areas. It's a dry net. So I think you have irrigated areas. Why is so important? Because when you put the microorganisms on the soil, in some days here, when we have dry lands, we have to produce below above the soil around 14 Celsius, 15 Celsius. It's very, very hot. For these microorganisms to survive the soil surface, it's very complicated. If you apply the fall we have a rain, okay, it works. But for rhizobium specifically, we need these ones in the root system. I think we have some perspectives about this. Okay, but the results is very depend. I think depending especially of the rain, because when rain, the organisms of spirillum, rhizobiums go where we expect they go for us.
If students have some questions, you can do it. Okay. So we are open for discussion.
Yeah.
Yes, Professor Buda wants to talk.
As our precision. How you are going to the data, big data, and so find the data, but data mining also concern more than the explain data.
Yes.
So I think it's okay. Next step is how can you in this system? So it's also same in my cases. So I also already develop new system to develop and predict the growth data strawberry. It actually has this. And also, so how can we send such the important and crucial information to the growers? So only sending the data has no sense. So we should send that as a meaning data. So how to do the how to think about this?
You asked about how we can do with big data.
Yes, big data is practical. So change the practical. Yes.
It's a challenge because we have different behavior and meteorological, and we have to find some prediction models to couple all of this. So it's a big challenge. I think working with people from computer programming would be the answer for that, because we know what we want to answer. I want to predict how many grams my chicken will grow in a day. I know the idea, but how to do the model is the big challenge. So I think we have some universities here in Brazil that also have specialization for big data. So I think we have to work with these people.
And statistical analysis for agriculture.
Yes.
It's very specific.
Yeah.
You should mention you predict what?
Everything.
You mentioned about chicken.
How many grams the weight the chicken will have today, tomorrow, based on the variation of temperature, light, everything we can consider to try to have a model to answer this. If you have, for example, there is a problem in the electric part of the farm, how this would impact the daily weight gain of my animals.
I have some comments on this. May I? Do you mind?
Can
Okay. Oh, I don't know, maybe you want to finish.
you want to finish first?
Yeah.
Okay.
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 Because by coincidence today I saw news in the internet that some producer from the western part was against Copel because they lost about 800 chicken yesterday. And due to some lack of energy, it's
really a common problem, you know. And once I was talking with some cooperatives, large cooperatives, and asking them about the way they pay the farmer is by the weight of the chicken.
Yeah.
Yeah. And not the way like bigger is better, not like this. They have a goal, an average goal,
Yes.
and if the weight goes much over than this, they reduce the payment, or even less than this average, they also reduce it. And there are some bonus to get close to
Yeah.
the average they decide. And I know that in the large 6 billion company, 10 billion company, who controls the weight? And they have to think, well, there are some guy here from TI, he made take some notes about this, and I said, but in the process, no control. And it reminds me the time I was in a teacher in the many years ago, teaching quality engineering or quality and productivity management. And there is no application of statistical process. Of course, that you would not have some mathematical model to predict the precise, but you can control the process by using statistics along the time. X, such kind of chart, medium amplitude, average amplitude chart. And I wonder that no one was applying that. You can every day control the evolution of the weight of the chicken. And as long as you have historical data, you have some inference about the weight of the chicken. You have another deviation associated with that, but it's a lot in terms of some variables that will affect not just the payment of the farmer, but all the process, the production process, the industrial process that we can later, because we have to adjust the process according to the change of the size and weight of the chicken.
That's why image analysis could be so important if we can estimate the weight of the animals.
Yeah, because you can make some sample of the process during the 42, 46 days of the chicken growth by applying weight control by image.
Yeah. For swine, we have some good examples for that. For chicken, the challenges.
First, they have such a big chicken.
Yeah.
But as much as 52,000 chicken per batch in everyone, and some of them they have 6 or 8, very close to produce 400,000 chicken every 46 days. So it's a huge number. And the worst also about such example, like beginning that the farm lost about 800 chicken, is that the farmer has to prove because of the sanity that the lot was caused by heat, not because of any virus or any other disease.
In Europe, we have some floor weight platforms. So the chicken go, you have how many chickens are there, and you have a weight measure. But in Brazil, not yet. So
it's easy to control. Or even if the operator, you can select a sample and check the weight one by one. Statistical, I don't know, maybe some every day how many chicken you have. But it's easy to make this kind of experiment.
Yeah.
But today, many of the main control of the weight growth made by the operator. And Of course, at the beginning, he wants to feed a lot the chicken, then the chicken, the legs open because of the.
Yes, the
and then
weight.
they have to-
Yeah. At most of farms, the person goes, takes some animals, waits,
Yeah.
oh, okay. We have to change this.
It's not a kind of predictive, but it's a kind of some statistical control. The capability of the process under with a very well known.
Yes.
One.
Okay.
So another big data. So is it possible that data that collected from the field and on the time because putting on open center. So as a maybe I think that some company in developing the stuff. And so far, made making it sensitive. Sharing I think it's really you are project.
Okay. Sharing.
Yeah.
We have to find some platform. All this data. Again.
Yeah, right. So we really compare. So in my case, about my data,
Yes.
because I is I engineer, but the academics is.
Yes.
I have two questions. Professor Dauri, and our regulations in order. How is our people here consuming? Yes. On agency of the body, and it knows that. But he's not doing it for. That even now the maximum amount of power is 50 games. It was is more out than 10 years ago. So it was you swing you this and also when you for some time for satellite connection even more restrictions. Like the part from Guadalajara, they check about this. Different index from processing content, try so we have one problem is that some meters are not. It is some years ago, strong epidemics of and it's more complicated. But overall, our regulation is really very clear
like the European Union. It's a more mandatory. In the United States, the regulation is advisory. It's a different and Professor Silas, I was wondering about this data about like line fixations. We work 6 places, it's a lot of work. I can imagine. Another thing that maybe the response in you said that there were no difference between work of 6 places, right?
Yeah.
And how of this was the same outward? Because maybe it can, we will have some interaction between my work, my conversation. Your opinions and the
Yeah, but the same. Yes, yeah. Because we think about this, yeah, but especially when we talk about the new ones and old ones have a great difference. But to not analyze our student is in same environment, because it's an important source of variation in this with the
understand. Different models.
Yeah.
Maybe in one location, but
Yeah. You know the good algorithm.
yes, it was right.
Yeah, it's put together in the scholarly. Yeah, really, yes. I know the it's very important.
I'd like to point out one more thing that about the maize corn production. Corn production has been a good support for Paraná, development of chicken meat industry. In the last 30 years, we increased a lot on production in the second harvest from March to July. And right now, Paraná is moving to ethanol production from maize. And that is a struggle. This B2G dry steel grains will be used in the exam for animal. So these are moving. That's what going on in Brazil. We are producing maize, and part of this maize will be used for ethanol.
The last question.
It's over.
Maybe you in the sentence. I mean, I've seen you presented basically that disease. What kind of disease? You guys seem
Many animal diseases.
Yeah, they you guys have studied here.
Here, you mean about the epidemiology?
No, which kind of disease are you guys studying here?
Here we are in Brazil, or here that I'm studying right now. Okay, no, yes, we are working in the animal science department.
But you have a pet school.
Yes, we have a vet school in Paraná. The vet is from State University of Paraná. And there they had some professors studying specific epidemiological factors to diseases and common diseases in Brazil, and also chronic diseases that animals and farm animals present. But specifically here in Maringá, we don't have that school. And I'm working specifically with dog and cat nutrition. But of course, I'm participating as an advisor also in the relay of sustainability and animal production on whatever. And there they have some researchers studying specifically clinical diseases and epidemiological factors. Okay.
Yeah.
One question.
Yeah.
Yeah.
You mentioned that the rainfall and soil affect the how about the variety? Variety of soybean?
Yeah.
I think it depends on the effect of the integration. Depends on the variety of soybean. How about the variety?
Yeah. You have beautiful results. This is not mine. Dr. Buranas, ele está prime, he's a professor in Piracicaba. Study about the effect of the, but basically the study of the Dr. Buranas was the year of the variety was released and effect on nitrogen fixation. So the study was performed in Guarapuava 2 years. Have some very, very important experience. But in Brazil, new ones rates fixation more nitrogen relative to the old ones. So it's important. Have more nodules, more nitrogen fixation. But we have some correlations between yield and nitrogen fixation too. Of course, the new ones rates is more productive relative to the old ones. If one of the cows is more nitrogen fixation. So for this one, for which I present here, we have the same variation. Yes, same variation, because it's a very important source of variation in trees. Okay.
So unfortunately, we have finished because we need to lunch and then go to CAP, and then we are going to Londrina. But all the discussions you can see on website, SETI website tomorrow and the other day. Okay, so I would just ask for everybody goes outside. We want to take a picture from everyone. Okay, thank you.